Hello everyone, welcome to the new session of plant tissue culture. So this is the second part of this topic, that is the plant tissue culture. Okay, so let us start this session. So let us talk about the callus. We have already introduced this callus. What is callus? So callus is defined as an unorganized tissue mass growing on the solid substrate. So this particular here in the picture you can see this is the solid substrate, that is the nutrient medium. We will study more about the nutrient medium in this session. Okay, so here we can see some unorganized tissue that is called as callus. Okay, so callus forms naturally on plant in response to wounding, infestations, or at graft unions. Its formation is central to many investigate and applied tissue culture procedures. So what happened when this is our nutrient media, and on this we put explant. And after proper condition, proper incubation, okay, what happened? This explant divide and form an unorganized tissue mass that is called as callus. So its formation is central to many investigative and applied tissue culture procedure. So our lots and lots of tissue culture procedure is based upon the formation of the callus only, right? Okay, so we are introducing here some of the terminology that we are going to use in the plant tissue culture. So first one is differentiated. Cells that maintain in culture specialized structure and function typical of the cell type in vivo. So there are so many cells that are used in the plant tissue culture. Okay, so those cells are cells that maintain in culture specialized structure and function. Those are called as differentiated. Explant, tissue taken from its original site and transferred to an artificial medium for the growth or maintenance. So the explant could be a part of stem, a part of leaf, bud, okay, any part from the plant. So that is the original site, okay, and it has been transferred to the artificial medium, that is the nutrient medium. So that particular part of the plant which we taken for the tissue culture, that is called as explant. Horizontal laminar flow unit. This is an enclosed work area that has sterile air moving across it. This we have already discussed in the previous session. Hormones. Growth regulators, generally synthetic in occurrence, that strongly affect the growth. So some of the growth regulators that are used in the plant tissue culture are cytokinins, auxins, and gibberellins. Internode, the space between the two nodes on a stem. So this is a stem and this is the node. And this particular part is the internode. So this particular part description is there in the plant tissue culture. So that is the internode. Media, that is the medium, nutrient medium, which we use to grow our explant. A nutritive solution, solid or liquid, for culturing cells or tissue. Micropropagation. In vitro clonal propagation of the plants from the shoot tips or the nodal explants, usually with an accelerated proliferation of the shoots during the subcultures. So here you can see micropropagation. That is the clonal propagation of the plant from the shoot tips or the nodal explant, whatever the explant we are using. Okay, so we are having a clone, okay, and lots of plantlets comes out from this. So this is micropropagation. Plant tissue culture, the growth or the maintenance of the plant cell, tissue, organs, whole plant in vitro. Okay, in vitro, in the laboratory. So that is the plant tissue culture technique. Regeneration. In plant cultures, morphogenation responds to the stimulus that results in the products of organs, embryos on whole plant. Okay. So here we can see in the figure explant. This is the explant. Okay. And this is the root part. And here we can grow a callus. So what we are providing here, CIM, callus induced medium. It means that we require a particular medium that can produce callus. Now this callus has been transferred to another type of medium. Okay, shoot regeneration. That is shoot-induced medium. Shoot-induced medium. It requires shoot-induced medium that is having high cytokinin and auxin in this particular medium, right? And when we want root regeneration, then we require root-induced medium, that is having higher auxin and cytokinin hormones. Okay, so in plant culture, morphogenation responds to a stimulus that results in the products of organ embryos on whole plants. So here we can, what we want to induce, according to that our medium is there. So we can do the shoot regeneration or the root regeneration according to the medium we provide. Soma clonal variation, phenotypic variation, either genetic or epigenetic in origin, displayed among the soma clones. So this is a very important topic we will study in one of the topic, that is the phenotypic variation, right? By using this particular tissue culture technique. Soma clones, plant derived from any form of cells culture involving the use of somatic plant cells. So we are using somatic plant cells, right? And the culture of those somatic plants gives you a type of plant that is having a different phenotype. That is showing the phenotypic variation. Right? So this is called soma clonal variation. We are using a somatic plant cell and that variate. There is a variation in the gene, right? And it shows the phenotypic variation. So this is soma clonal variation, and the cell which we are using, somatic cell which we are using from the plant cell, that is called as soma clones, or the plant derived from the cell culturing involving, that is the by use of somatic plant cells. So those culture, those plantlets derived from those culture is called as soma clones. Sterile. What is the meaning of this? Without life. Or it is another meaning that inability of an organism to produce functional gametes. So the organism that cannot produce the functional gametes are also called as sterile. But in the case of plant tissue culture, a culture that is free of viable microorganism, okay, no organism is present, then we can say that culture is sterile. sterile techniques. From this sterile, we are having a term sterile techniques. The practice of working with culture in an environment free from microorganisms. So we always require sterile environment for tissue culture so that our culture do not get contaminated with the microorganism. So this technique is also called as sterile technique. Totipotency, a cell characteristic in which the potential for forming all the cell types in the adult organism are retained, right? So root of a carrot plant is taken and the transfer section, you can see it is cut from here and you can see this transfer section, right? So we can have a fragments of this root part, fragments cultured in a nutrient medium, right? A free cell in a suspension begins to divide. So in this suspension, what happened? The cells from this tissue begins to divide in this suspension. So now embryoid, a somatic embryo develop from the cultured free cells. From this free cells, these are the free cells that develop an embryoid. And from this embryoid, we can see whole plant. It is having roots, it is having shoot even, and we can see this adult plant. So there are some cells that can produce all other types of cells, in the plant body. That particular cell is called as totipotent cell. And this phenomena, that is from one particular cell giving rise to a whole plant, that is called as totipotency. Okay, so this is very, very important. Undifferentiated. With plant cell existing in a state of a cell development characterized by the isodiametric cell shape, very little or no vacuole, a large nucleus, and exemplified by cell comprising an apical meristem or the embryo. Nutrient media. So we will now discuss about the nutrient media in which the explants or the microorganisms are cultured. Okay, so for this culture we require medium, right? And in this medium we require some inorganic nutrients, organic nutrients, hormones, right? So let us study about what are those nutrients that are present in the culture media. The first one is the inorganic nutrients, which includes macronutrients like nitrogen, phosphorus, potassium, calcium, and also the micronutrients like boron, copper, iron, manganese, zinc, and etc. So these are the macronutrients, like it means that they are required in a larger quantity: nitrogen, phosphorus, potassium, and calcium. But there are some elements, okay, some inorganic nutrients that are required in a small quantity, but their importance, they are very, very important: boron, copper, iron, manganese, and zinc. Organic nutrients. It includes vitamins like vitamin B1, B6, B3, and B5. Okay, some amino acids are also present in the nutrient medium as an organic nutrients, that are arginine, asparagine, cysteine, and glutamic acid. Okay, for carbon source like glucose or maltose is required, and some growth hormones or the regulators like auxin, cytokinins, gibberellins, ethylene, and abscisic acid are also present as an organic nutrients in the nutrient medium. And other media substances like protein, hydrolysates, yeast extracts, fruit extracts like banana fruit extract has been used, coconut milk, solidifying agents like agar, alginate, gelatin, etc. are required. Some iron sources also required like EDTA, that is ethylene diamine tetraacetic acid. So this is used as in the treatment of heavy metals. Why it is used? Because it is used as a chelating agent. Now, what is the use of a chelating agent? It binds to the calcium. This EDTA binds with the calcium. Okay, it binds with the calcium and prevent the calcium to bind with the cadherin proteins. Cadherins proteins that are present at the surface of the cell and do not allow cells to forming the clump. Okay, do not allow cells to forming a clump, and now the cells are present individually in the suspension medium. So for this iron source, EDTA is required. Okay, pH of the medium should be in the range of 5 to 6 before autoclaving the culture medium. So here are some of the essential elements. Nitrogen, component of protein, nucleic acid, some coenzymes, element required in the greatest amount. So nitrogen must be present in the nutrient medium as an inorganic nutrient. Same with the potassium, that regulate the osmotic potential and the principal inorganic cation. Calcium, that is very much important for the cell wall synthesis, membrane functioning and cell signaling. So this must also be present in the nutrient medium as an inorganic nutrients. Magnesium, that work as a coenzyme cofactor, component of the chlorophyll. Phosphorus, component of the nucleic acid, energy transfer, component of the intermediates in the respiration and photosynthesis. Sulfur, component of some amino acids, that is the methionine and cysteine, that is the sulfur containing amino acids, and also some cofactors, they require sulfur. So these elements must be present in the nutrient medium. Chlorine, required for the photosynthesis. Iron, electron transfer as a component of the cytochromes. Manganese, so these are some of the elements that are required in a less amount, that is a micronutrient. Manganese, enzyme cofactor. Copper, electron transfer reactions. Zinc, enzyme cofactor and chlorophyll biosynthesis, they require for this. And also molybdenum, enzyme cofactor, component of the nitrate reductase enzyme. So here are some of the plant hormone. Okay, the plant growth regulator used in the plant tissue culture media, normal concentration, that is in the range of 10 to the power minus 7 to 10 to the power minus 5 m. P-chlorophenoxyacetic acid, 2,4-dichlorophenoxyacetic acid, indole-3 acetic acid, indole-3 butyric acid, naphthalene acetic acid and naphthoxyacetic acid. So these are the different forms of oxen and their abbreviation is also given here with their molecular weight, and all oxen dissolved in dilute NaOH or the aqueous ethanol. So according to our culture medium which we are preparing, we require oxen. So any of the oxen we can use, right? Cytokinin, 6-benzyl aminopurine, N-isopentenyl aminopurine, 6-furfuryl aminopurine, that is also known as kinetin, and the zeatin is also there. So their abbreviation is given here and their molecular weight is also given here. And all the cytokinin are dissolved in the dilute NaOH or the aqueous ethanol. So there are number of cytokinin that are available. We can use anyone according to our culture medium we are preparing. Gibberellin and abscisic acid. Gibberellic acid, that is the Ga. There are a family of the gibberellic acid, right? So dissolve in water. Gibberellic acid and abscisic acid ABA. So they are dissolved in the aqueous ethanol medium. Macro elements: ammonium nitrate, magnesium sulfate, calcium chloride. Okay, these are the macro elements. Some of the micro element: H3BO, CaCl2, copper sulfate, nicotinic acid, pyridoxine, thiamine. Okay, so these are some of the organic compound that are required. Okay, and this is called as a Murishik-Skog medium. This particular medium, we require these macro elements, micro elements, and organic compounds. KPH2O4, KNO3 are also required for this as a macro elements. ZnSO4, Na, MoO4, MnSO4. So there are so many micro elements that are required, okay, for this particular preparation of this particular Murishik and Skog medium, that is also called as MS medium, and that was developed in 1962. Okay, so this is all about this session. We will have one more session for the plant tissue culture. Till then, thank you very much.